Okay. Hello, everyone. My name is Kondeti, and I am a PhD candidate in the Langert Lab at Loyola University, Chicago. And I'm really excited to share my thesis work with you today about targeted drug delivery strategies we are working on for Charcot-Marie-Tooth disease, specifically CMT1X. So through the conference, we've seen so much exciting and promising research, including innovations such as small molecule and gene therapies. But one thing that's been consistently presented as a challenge for moving these treatments forward in the field is the difficulty of getting past the blood-nerve barrier to administer a localized treatment. And so what we are proposing in the lab is to look at the state of the blood-nerve barrier more closely and to figure out a way to harness the pathologies that we see within CMT1X to create a targeted delivery solution. And so as I mentioned in the last slide, one of the biggest challenges for administering a localized treatment is the blood-nerve barrier. So the blood-nerve barrier is an anatomically restricted site. It's a tight barrier that prevents the free entry of molecules into the endoneurium. And so what we are proposing as a solution is targeted nanoparticle delivery strategies. And so through this talk, I will tell you about the pathology that we have found within the peripheral nerves in Connexin32 knockout mice, particularly regarding immune infiltration and blood-nerve barrier integrity, and how we plan on harnessing these pathologies to bypass the blood-nerve barrier to provide our targeted treatments. And so in order to create a targeted treatment, we're looking to harness pathologies within the disease that can naturally bypass the blood-nerve barrier, such as immune infiltration. And so immune infiltration has been widely characterized in many different forms of CMT, including CMT1X. And so I can show you here that infiltration has been shown to begin at 3 months of age, progressing through 6 months. And this coincides with an upregulation of the inflammatory chemokine CCL2 across the nerve. And so in order for us to get a better idea of how we could harness this immune infiltration, our first step was to build an immune infiltration time course so that we could particularly focus on some earlier time points. And the point of this was that we wanted to find the best window of opportunity to administer a treatment that could potentially allow us to give something like a gene therapy early on before the onset of the disease to hopefully prevent its development or stop it entirely. And so here we've replicated what literature has shown previously with a robust infiltration at 3 months, progressing through 6. But what we really noticed was this cliff at 3 months of age where infiltration really seemed to be ramping up. But we found this suggestion of macrophage infiltration beginning at 2 months of age with IHC, which we can see in the quantification as well. And so we wanted to dive a little bit deeper in this, but we found that using immunohistochemistry presented a little bit of a limit with how many cells we could see, and we weren't able to see a true full picture of the nerve. And so in order to get past this, we wanted to utilize flow cytometry and look at whole nerve homogenates for total cell populations. And so here I have some representative graphs of wild type versus knockout 6-month data. We're looking at here CD45 and CD11b double positive macrophage populations. And so here, when we look at the 6-month data, we see a nice, robust presence of these double positive macrophages in the knockouts, and we don't see them in the wild types. And so this has been shown in literature, but when we looked at the earlier time points, even as early as 1 month, we were able to find macrophage infiltration in our knockout nerves versus in the wild types, which we were really excited about. And so here I have some sex-combined data of our flow cytometry results versus our IHC results. And so there are a couple things that we can really appreciate from this data. First of all, we can see the scale with which we're able to see the number of macrophages in the tens with IHC versus in the hundreds with flow cytometry. And then when we dive deeper into the 1 and 2 month data, we can see that there is a significance at 1 and 2 months of age when we look at the number of macrophages in knockouts versus the wild types. And then when we break this data down by sex and separate it out, we also see a separation of the significance, where we see significance as early as 1 month in males and as early as 2 months in females. And another thing I really wanted to point out here was the difference in scale of immune infiltration between the males and the females. We can see that the females appear to have a greater macrophage infiltration than the males do. And so then our next step was to look at BNB integrity more closely. And so our first way to do this was utilizing our flow cytometry. We also looked at CD31 and CD54 double positive inflamed endothelial cells. So when we look at 1 and 2 month data in here, we don't see significance of these inflamed endothelial cells, indicating that there may not be as many changes going on in the pathology of the blood-nerve barrier at the same points where we're seeing significant infiltration. And so then when we utilized Evans blue in order to look at the permeability of the blood-nerve barrier, when looking at wild type versus the knockout, we did not see the striking presence of Evans blue within the nerves. So to orient you to what you're looking at, we have longitudinal sections of nerves where we can see CD31 along the endoneurium, and these are the labeled blood vessels. And so you don't see that striking Evans blue. And so to show you what characterizing blood-nerve barrier permeability with Evans blue could look like, I have a comparison that we have in our lab in our experimental autoimmune neuritis model, where you can really appreciate between control and the EAN, this bright presence of Evans blue indicating blood-nerve barrier breakdown, which we don't see in the CMT mice. And so this, combined with our inflamed endothelial cell data, really showed us that there may not be many changes occurring to the blood-nerve barrier, that it may not be becoming permeable at these early time points. And so to create our targeted delivery strategy, this showed us that we wanted to take a more active targeting approach that would allow us to infiltrate through the blood-nerve barrier. And so our first working hypothesis is that CCL2 drives early immune infiltration in CMT1X. We can see that there is significant infiltration at early time points, and we know that CCL2 is upregulated in the nerves. And so in order to create a delivery system that can be actively recruited into through the blood-nerve barrier into the endoneurium, we wanted to look for a way to coat the infiltrating immune cells onto our nanoparticle cores to create a targeted system. And so this has been done in literature before. So I have an example from a paper by the Lang group in which they were able to take macrophage membrane vesicles that were enriched with their desired targeting ligand, coat them onto nanoparticle cores, and then direct them towards inflamed vasculature in a rheumatoid arthritis model. And so in order for us to create a similar system, the first thing that we needed to do was figure out a proper cell source that could contain CCR2 on its surface so that we could direct towards the CCL2 seen in the nerves. And so when looking in literature, we found that monocytes, rather than macrophages, which we've seen as the immune infiltrate, are actually the infiltrating bodies and are enriched with CCR2 on their surface. And so following this, we're able to take a 3-step method where we can isolate plasma membranes, coat them onto our nanoparticles, and then administer them. So first we have the plasma membrane isolation, where we're able to show that we can get plasma membrane fractions that are enriched with our desired targeting ligand. Next, we create our nanoparticle cores that have a quantum dot stably labeled bright fluorophore integrated into the core, allowing us to image these via SIM imaging. And then we can also load them with a dye to mimic a payload. And then finally, we are able to coat the nanoparticles with these membranes that we have harvested. And here I have an in vitro assay where we were able to show that these coated nanoparticles would travel preferentially towards a condition that contains CCL2 versus one that does not. And so then we moved this in vivo in our mice. And so in order to do our mouse injections, we do tail vein injections, allow the mono-NPs to circulate for 24 hours. Then we perfuse the mice and then harvest organs for ex vivo imaging. And so first I'll show you imaging that we did via structured illumination microscopy, or SIM imaging, where we can appreciate the presence of our mono-NPs within the males in the vessels and in the females into the endoneurium. And notably, we don't see the same presence of these nanoparticles in the wild types. And then when we look at the ex vivo data, we can really appreciate, first of all, in the females, the presence of our mono-NPs in the sciatic nerves of our knockouts, where we don't see them in the wild types. And a very interesting sex difference that popped out here was that while we did get infiltration into the female sciatic nerves, in the male knockout nerves, we did not see that same kind of infiltration. And so here I have the quantified data. Again, we see significant infiltration of the mono-NPs into the sciatic nerves of our female mice, and we don't see that same infiltration in the male mice. And so overall, we believe that targeted drug delivery strategies, such as the one that we are working on in the Langert lab, are the future of the CMT field. These strategies will allow us to provide targeted treatments to the peripheral nerves at earlier time points to allow us to reduce any off-target side effects and also administer a gene therapy early. And so I'm really excited to see this work continue to evolve, and I hope that we can utilize these strategies across multiple forms of CMT in order to improve patient lives. And so with that, I just want to say thank you to my lab and of course to the foundation for funding our work and for giving me the opportunity to present it today. Thank you.